cycle of it. It starts small and gets very, very complicated. And this structure here is what we're interested in. That's where the exchange takes place. That's where the plant dumps off the phosphorus in the water and receives payment in form of sugar. We took some corn roots that were colonized by these fungi, looked at them with the X-ray microscope, and you can see in the red box there's one of those arbuscules, and this is one slice through the volume, and I did an animation on the right-hand side. And you can see a number of these arbuscules sort of alternating in the pavement stone cells of the roots. So they're all over the place. You can even see some of the fungal strands that connect these two. These are hyphae that will sort of connect the various feeding structures on the inside. Cool imaging, so what? Bring in knee. 3D volume and mapped the entire thing. So everything in yellow is a fungus.
Wow.
So you can see the long stripes where the fungus is connecting, all the different arbuscules, and then the roots are kind of in the background transparent. We can now measure the entire fungal colony inside these roots. So this is, again, a way that we're trying to understand how these organisms interact so that we can encourage this in as many situations as possible. Again, let's revisit the fact that corn roots are incredibly diverse. Architecture is amazingly complicated, but roots don't do much. They'll either just elongate, they'll bend, or they'll branch. That's all they can do. But they get signals, genetic signals, environmental signals, to do these in very, very different ways. And you can see the huge diversity. Our question right now is how early can we detect differences? So, Nee, it's a broken record. She's brilliant. She wrote a paper last year that looked at corn growing in a gel, and she looked at it from sort of day 6 to day 11 and mapped the whole thing. And you can see these two different corn lines, B73 and Missouri 17. They're simply different corn lines. They're very, very different architectures. And we can see that as early as 11 days. We plant those out in the field, we collect the roots, and we scan them with the X-ray. And they're also very, very different, even at maturity. And with multivariate statistics and a very, very complicated looking graph, the take-home message is all the yellow symbols on the left are very, very different than all the aqua or blue symbols on the right. Meaning that it doesn't matter what age we looked, whether it was young plant in the gel or old plant in the field, the roots that we could measure with X-ray were very, very different. And we could look at this, we could probably know the answer, but we have to prove it. We have to do it statistically, and we have to get it published, which means evaluated by our peers and approved. That's the power of science.